This is Biology 1, Chapter 12, Part 3 on molecular genetics, or DNA. In this video lecture, we will be discussing the process of DNA replication, which, remember, occurs during the S phase of interphase during the cell cycle. So at this point in the journey of DNA, we have discovered the function of DNA, which is the genetic material and instructions to pass on to new cells. And we now understand the structure of DNA, which is the sugar and the phosphate are in the backbone, the nitrogenous bases are in the middle, and those bases are connected by a hydrogen bond. Now, Watson and Crick did hypothesize about how new DNA gets into a cell. And the idea was, since we have this complementary base pairing, A's equals T's and C's equals G's, there is a way to get the same exact instructions in every new cell using this complementary base pairing method. And the idea was that one strand of new DNA would be made from a strand of the original or parental DNA. And the parental DNA would be used as a template to put the same exact information into the new cell. That won't work. So the idea of semi-conservative replication is that there is one strand that is always carried over into the next cell. And because we have complementary base pairing, then if, for example, the parent strand, you can see the top strand and the bottom strand are paired complementary. A pairs with T and C pairs with G. Now, if I take the top strand, which reads ACT, CGC, CTA, and I use that as a template in the first replication, guess what I will get for my other side of my DNA molecule? The same sequence that I had in the original parent strand. If you semi-conserve, or if you keep part of the DNA molecule to be used in the new cell, you'll always get the same sets of instructions for every new cell that is made. And you can follow along in this diagram. If you'll notice, the top strand and the bottom strand, no matter what color it is, it's always the same sequence of DNA molecules. So in order to replicate DNA, we got to get into that molecule. Remember, the DNA is in a double helix. It is a wound up molecule. So the first thing we need to do is unwind and unzip the DNA double helix. There's a special enzyme involved in that, and it's called DNA helicase. DNA helicase breaks the helix. That's why I call it DNA helicase. So first, that enzyme comes down, unwinds the DNA molecule, and then it breaks apart the hydrogen bonds during that unzipping process. Then, in order for the rest of the enzymes to know where to start the DNA replication process, another enzyme that's called RNA primase comes in. RNA primase adds an RNA primer, or a start point, on each DNA strand, so that the next enzyme that comes along knows where it's supposed to begin this process. The next enzyme that is involved is DNA polymerase. Okay, a polymer is a large molecule. That's what DNA is doing. It's adding nucleotides to the new DNA strand. So it's using that semi-conservative replication. So if DNA polymerase is standing on an adenine and looks across the DNA molecule, it's going to put a thymine. If it sees a cytosine, then it's going to put a guanine, and it'll go on and on and on. The leading strand which is the one that goes from 3' to 5', replicates normally. DNA polymerase keeps adding on bases until it's done. DNA polymerase only works in one direction. So the bottom strand, remember the 5' to 3' strand is backwards from the leading. So the lagging strand has to replicate backwards in little sections because DNA helicase only works in one direction. The little goofy backwards pieces that are made on the lagging strand are called Okazaki fragments, named after the pair of biologists that discovered this process and how DNA polymerase adds the nucleotides to the lagging strand. So once we have finished our DNA replication process, once we've unwound and unzipped the molecule, once we've added a primer, once we've added all of our nucleotides, then we have to wind and seal up our DNA molecule. The enzyme DNA ligase is responsible for that. DNA ligase is responsible for connecting the two sections of DNA back together. So DNA ligase adds the hydrogen bonds. DNA helicase breaks those bonds. Now, there is a difference in how this is attempted in both prokaryotic cells, which are cells without a nucleus, cells such as bacteria, and eukaryotic cells, which are cells that are in animals and plants. Pardon me for that interruption. Animal and plant cells are eukaryotic. They're more complex. Okay, eukaryotic DNA and prokaryotic DNA. Think of DNA replication like road construction. If you were going to fix the entire Interstate 90, all the way from Boston to the West Coast, wherever I-90 ends up, I can't think of off the top of my head, but it's an expansive highway, goes across the entire nation. If you were to fix that highway, you wouldn't start on the East Coast and have one crew working until it finished to all the way to the West Coast. It would take way too long. You would have multiple construction crews along the way fixing sections of the road. That's how eukaryotic DNA replication works. This is going on in multiple areas along the DNA strand. So there's many construction crews unwinding the molecule, finding a start point, adding the bases, and sealing it back up. Prokaryotic DNA, on the other hand, would be like fixing Main Street in Casson. You could successfully start with one construction crew at the beginning of Main Street and with one crew work all the way to the end of Main Street because it's a lot shorter. Prokaryotic DNA, remember, they have their circular plasmids, and basically they start at one point on the strand and they go all the way around and replicate until they finish. And of course, the top and the bottom strand in the prokaryotic DNA is named after Watson and Crick. So we have the Watson strand and the Crick strand for prokaryotic DNA. So think of it like road construction. Eukaryotes, complex, lots of repair along the way. Prokaryotes are very simple, so they can start at one point and go all the way to the end and finish. So the enzymes involved in DNA replication. We have DNA helicase, unwinds and unzips the DNA molecule. It breaks the hydrogen bonds. RNA primase gives a primer or a start point so that DNA polymerase can come in and add bases. Once that's completed, DNA ligase winds the molecule back up and adds those hydrogen bases to zip the double helix structure back into place. And this has been part 3 of chapter 12 lecture on molecular genetics and DNA.